Okay, this is where we left off on the last one, and I found my information with regard to these numbers, and I was off in the human cell. Listen to this. Human cell can copy 6 billion base pairs and divide into daughter cells in only a few hours, and all of that with only 1 error per billion in nucleotides. That's just amazing. But one of the reasons why this is it's so imperative that that the nucleotides be exact is because just one base pair off, then which we will learn later, the base pairs are made in triplets that can actually put one whole 3 areas of nucleotides off, and that those errors actually amplify as you reproduce that molecule over and over again with RNA. So we learned that RNA goes into the nucleus and then makes a template for the DNA, but if that template is wrong, then when it goes out to create the protein in the cytoplasm, then that protein will be all off. And some of these proteins are highly important with regard to normal functioning, and some of them aren't as important, but all of them are made for a purpose. But just it's mind-boggling how many nucleotides base pairs are replicated and how few errors there are. But next, the next idea is, how does this happen? And so that evolved over time. One of the first things that was discovered, again working with bacterium that tends to be a little simpler to study, is there's this origin of replication, and you can see this in the left portion of the image looking at the E. coli bacteria, that there's a definite spot where replication begins. It happens to be a group of a specific sequence of nucleotides that are recognized by enzymes. We're going to talk about a few enzymes and a couple of proteins, but there are something like 130 that have been recognized. I'm trying to look at my number on this, about 130 that's been recognized in humans with regard to replication, but we're only going to talk about a few of them here. So you get the general idea of replication. But what happens is there is a certain sequence of nucleotides, and you see just one in the E. coli bacteria, but in eukaryotic cells there are multiple throughout the long line of the chromosome. So the chromosome is replicated very fast because it's not just starting at the beginning and going to the end. It's starting in these various different areas. So at these origins of replication, what happens is enzymes separate the strands, forming what's known as a replication bubble. And so you can see a little bit of a bubble. We're looking at the second circle over from the left in the bacterium. That then goes down into a larger and larger circle till it ends up replicating. So you have that circle becoming bigger and bigger until it becomes the size of 2 cells, and then those cells then separate in binary fission, and then you have the duplication of the bacterium. What happens here in the eukaryotic cells, and you can see images at the bottom, the X-ray image, or at the right where the eukaryotic cell, and the little fuchsia arrows are pointing to bubbles of replication, the origins of replications that have turned into these bubbles because of enzymes have separated the nucleotides, and then the chromosomes, I mean the nucleotides, can then be added at these replication forks. And so what you have, we've just taken the previous image and just looking at a little piece of it. You have your replication fork there. So the DNA has been separated. you have your replication fork there, and one of the things we want to add is this idea of the 3' and the 5'. You do have a hydroxy end, and that hydroxy end, which is the 3', hydroxy, remember, is that OH negative that has a hydrogen and an oxygen. This nucleotide base is going to be attached to the hydrogen. The oxygen is going to come off. So the actual adding of the nucleotide bases has to be on the 3' end. 5' is where you have a phosphate. You have a phosphate there. So you have a different ending to each of the parent cells, or the parent ends sides of the DNA. But the other things that you have on here, you have your replication fork. This is being caused by this bubble, and you have other enzymes. You have this helicase, and helicase's job is to unwind the DNA, so kind of unzip it, you might say. And this topoisomerase its job is to actually unwind it, so make it straighter, and then the helicase comes in and unwinds the nucleotide bases. What the single-strand binding proteins do is stabilize it, and stabilize it in a couple of ways. Not only make it less fragile, I guess, as a piece of DNA, but also keep the nucleotide bases from getting back together. It's only natural. They are, remember, they're attracted to each other. Those purines and pyrimidines, because of the hydrogen bonds, they're attracted to each other. So this single-stranded proteins keep them apart, and also just stabilize the fragment itself. The primase is actually an RNA primer that gets put in there, so that then the enzyme that's going to bring the new nucleotides in recognizes where it goes. Primase basically marks it. So DNA polymerase can't initiate, that's the enzyme I was talking about, can't initiate a polynucleotide, the synthesis of the rest of the strand, but only add nucleotides at the 3' end. The initial nucleotide strand is that short primer. So that short primer is attached, then the polymerase, DNA polymerase, which is bringing in the nucleotides, is going to recognize that primer. That primer is put on the 3' end. That primase, sorry, let me use the right terms. It starts an RNA chain from scratch, and adds RNA nucleotides one at a time, using the DNA as the template. It's a short 5 to 10 nucleotides long, and that marks the starting point for a new DNA strand. Then with regard to further understanding about what happens, this is where you have this concept of the use of DATP. We are familiar with ATP, and we're familiar with ATP, what it does for us with regard to providing energy because of those phosphate tails. So we'll just kind of refresh our memory about it. Is you have this is deoxy. Deoxy instead of ribose, so deoxy is the sugar with DATP, and so that makes it a little bit different. And I apologize in advance if I don't say DATP, but that's what I'm referring to. But you do have these 3 phosphates, and the molecule with the 3 phosphates with a sugar, and you also have a hydroxy active unit hanging off, and then you have your nucleotides. So what you have, if you look at the picture on the left, the the left portion of the picture on the right, I guess you might say, but you look at those 3 phosphates bringing in the sugar and then also the nucleotide base. It tends to be thymine. It's going to attach to adenine because of those hydrogen bonds. And what actually is going to make it attach to the unit above it, that GC unit above it, is you're going to have a pyrophosphate made. So what's going to happen is 2 of those phosphorus are going to come off, and in pulling those off, you're actually creating energy. So this is an exergonic hydrolysis, is what the hydrolysis. So an exergonic hydrolysis reaction is actually going to put that nucleotide base on the growing line of the new template strand. So that's why the new bases can only be added on the 3' end, because that's the area that has the hydroxy group hanging off. We're going to attach the sugar to the hydrogen bond, take the oxygen off, put that together with 2 of the phosphates, and then you're going to have the oxygen and phosphate group come off, and then the new sugar is going to attach to the hydrogen, and then the thymine is going to attach to the adenine via hydrogen bonds, and you're still going to have a hydroxy group hanging off the other end that allows the next nucleotide to be base to be added. And so this is how DNA polymerize. This is how this works. This is an enzyme that is actually going to make these long chains of nucleotides, these long units, one right after the other over and over again, from the 3' to the 5'. So from the 3' to the 5'. Each monomer of DATP joins with the DNA strand, and it loses 2 phosphate groups as a molecule of pyrophosphate. There you go with regard to how that happens. Each individual nucleotide gets added. I just added this particular slide. It might help you a little bit to understand what each of these do. Remember, the helicase unzips it. The topoisomerase straightens it out. Primase synthesizes the RNA primer at the 5' end of the leading strand and at the 5' end of the Ogasaki fragment of the lagging strand. We haven't talked about that yet, so don't freak out if you don't remember that. And then you have these polymers, polymer 1, polymer 2. They are acting, we're kind of putting them all in a new group. They synthesize new DNA by adding nucleotides. You do have a little bit different. Polymer 1 removes the RNA nucleotides from the primer end. So it removes nucleotides. DNA polymer 3 actually adds them, but I won't make you distinguish those two. And then ligase, which we haven't talked about yet, but it's good to know this a little bit. What that does is joins the strands, the Okazaki fragments, joins them together into one. So let's learn a little bit more about what's happening. This happens because remember you have 2 separated backbones. And this is happening in, both of them are happening at the same time in these various origins of replication, the bubbles that have been made all the way along the entire length of the chromosome. And so DNA polymerase adds nucleotides to the free 3' end, and then can elongate only in the 5 to 3 direction. So it's only going to be able to elongate in one direction. So from the parental DNA from the 5 to 3 direction. Along one template strand of DNA, the DNA polymerase is the leading strand and continuously moves towards the replication fork. So you have the leading strand, and then what you're going to have is the leg